Hello students, welcome to Leela's tutorial. Today's topic is embryo culture and embryo rescue. Right? So here you are going to culture the embryo in order to produce a new plantlet. Okay? So here the normal embryo can be produced in vivo also. Right? That means by natural process it will germinate and give rise to plantlet. So why we have to go for this embryo culture? Because in case if we want to produce any new variety, right, new species, by combining two species or different plant species, you can produce a new one which cannot be done by natural process, right? So in that case here the scientist Hannings is the first person in 1904 who was successful in producing plantlets by embryo culture, right? He has selected two different plant species. From first plant species he has taken the male gamete, that is the pollen grains, and another plant species he has taken the female gametes, that is the ovules, right? Then after successful fertilization of these male and female gametes, it resulted in formation of zygote. Then zygote developed into an embryo, and then embryo by means of plant tissue culture, he was successful in producing a new plantlet, right? So here let us see if it is a normal seed. Right? In the normal seed, the seed is protected with the outer hard seed coat. Right? Then here this is the embryo which may give rise to a new plantlet, the shoot and the root system. Right? And the endosperm is a nourishment to this embryo. It provides all the nutrients required for the growth and development of the embryo. Right? So here we are going to culture this embryo. So let us see in detail. First, it is the embryo culture which is differentiated into two types. Okay, first one is mature embryo culture and second one is immature embryo culture. In normal fruits, if there are viable seeds, what happens when they are sown in soil, they will give rise to plantlet. Right? So that means they all are mature embryos. So culturing these mature embryos is easy. But here in case of immature embryo culture, that means embryos are immature. Okay, so when you sow them in the soil, they may fail to germinate because of partial development, right? So what you have to do, you have to save these immature embryos and go for your embryo culture so that you can produce your plantlets. So here that is nothing but embryo rescue. You are saving or you are protecting the embryo from damage or degeneration, right? So you are not allowing the embryo to die. You are rescuing it and you are allowing it to produce, develop into a new plantlet. Is that clear? So let us see the process. Here first you are going to take a fruit, right? Then you are going to surface sterilize it by using your sodium hypochlorite for 10 to 15 minutes. Then rinse with double distilled water so that no remnants of sodium hypochlorite will be present. Right? If any remnant of this chemical is present now, then again it will interrupt in the development of your embryo. Okay? So after rinsing with double distilled water, you are going to air dry them in sterile Petri plate. Right? So in a sterile Petri plate, allow it to air dry. Place everything all set up inside the laminar airflow chamber. Okay? Then cut open the fruit under sterilized conditions with the help of a scalpel and the forceps. Right? You are going to extract or take out the ovules or the seeds from the sterilized fruit. Okay? So once you collect these in Petri plate, either you go for in vitro, it is a natural process where you sow the seed so that it will germinate on its own, right? But if it is an immature embryonic culture, so what happens if it is an immature, it fails to germinate naturally, right? So the in vivo will be absent. So what happens? You have to save this embryo. If there is no germination, gradually that embryo will be lost, right? You doesn't want to lose that and you want to produce a plantlet from that embryo, right? So here what you do, you select in vitro process. In vitro means in the lab. You are culturing in vitro, that means you are culturing the immature embryos in the lab by providing the favorable conditions, nutrients and other compounds required for that embryo rescue process. Right? So once you do that, here the embryos will be matured and you can go for your embryo culture process. Okay? So now you can see, once after fertilization, you have got the embryo. Now the zygote which is present, okay, that will develop into an embryo. The immature embryo will be matured, right, in plant tissue culture media under favorable temperature, pH, nutrients and all. Then you can see it gradually develops into heart-shaped embryo. Then the heart-shaped embryo, after incubation for a certain time, it elongates, right, and a tail-like structure appears, which is called as torpedo-shaped embryo, followed by cotyledonary stage. So here it is same as the cotyledons of the natural seed. When you sow the seed in the soil, first the cotyledons will be observed, right? So here also it is the cotyledonary stage. So after germination of this, everything is occurring in plant tissue culture media. After germination, you get the shoot and root system with required auxin to cytokinin ratio. Right? So like that we can produce a new plant species by crossing or by taking the gametes from two different plant species. Is that clear? So if you have any doubts, put it in comments.